I'm going to wait until I get a signal from Mario to introduce you because I don't know when he wants to start exactly.
That's fine. How's the weather there, Rick?
A little bit rainy and cold, not like SoCal.
People here start complaining when it gets to 60, it's like, oh, it's cold.
Yeah, now they won't be complaining. It's just par for the course. But how are you doing?
I'm,
Do you have
I'm,
a good
I'm,
Thanksgiving?
yeah, it was a great Thanksgiving. How about you?
Good. Yeah, we get together with family in Southampton, so we had a big group out there. Uh, I live in the city, but we usually get together. We have a place in Southampton. We get together the whole family out there.
Sounds nice.
Going from one to the other and back to the other. Good morning, good morning. Can you hear me, Rick? No.
Yes.
I am sorry there's some audio here, seems to be some reverberation, so I'm gonna have to kill the other microphones after you.
Should I
So,
go ahead and introduce Dr. Marbán? Mario? I'm going to assume that's a yes, and the other
It's a
microphone.
a delay.
Okay. Well, it's a real honor and a pleasure to introduce Dr. Eduardo Marbán, who's a friend and a long-term colleague of the last more than 30 years, longer than either of us, I think, want to think about. Eduardo is originally from Cuba, came to the United States, and was in the MD PhD program at Yale. It's interesting, his first paper at Yale, the first paper he published was on measuring calcium in contracting heart cells. And it was done with the Chen brothers, Roger, who won the Nobel Prize, and Richard, who was his PhD mentor. He then did his residency and cardiology fellowship at Hopkins, eventually became the chief of cardiology at Hopkins. And Hopkins has this magnetic draw to people. They almost never leave, but he was a renegade. And it didn't surprise a lot of us because we knew that when he picked up and left and went to Cedars-Sinai. And at that time, Cedars-Sinai was a very good hospital, the hospital of the stars, as they say. But he made it into like a #1 cardiology place by running the whole operation, cardiology, cardiac surgery, the research and everything. And the rest is history. So I'm going to leave the intro at that and leave it to Dr. Marbán to discuss discovery arc from cell therapy to novel RNA drugs. Eduardo.
Thank you, Rick, for that kind introduction, and thank you, Leandro, for the invitation. I should note that Leandro was one of our stellar fellows, and we're super proud of how well his career has gone at Montefiore. So in talking today, I could give you the fantasy version in which we started working on cell therapy for heart disease. And through predictable and iterative steps, we ended up creating new RNA drugs for a lot of intractable diseases, some of which ostensibly have nothing to do with the heart. But it really happened in a different way, a circuitous path in which each inflection was marked by a discovery, a mechanistic discovery that caused us to change direction, reassess what we were up to, and realize that we had new opportunities that weren't there even as recently as the most recent inflection. So the story begins at Johns Hopkins a couple of decades ago when I first started creating a cell product that's now called deramiocel. We gave it a process-derived name, cardiosphere-derived cells. But these are basically cardiac stromal progenitor cells. They fulfill the criteria for being progenitor cells insofar as they're multipotent and have some stem cell-like characteristics, but they don't work that way. The central panel shows that basically the way they work is by
Sorry, Dr. Marbán, can you hear me?
Yes.
So I wonder if you have a speaker room, place your microphone, turn off the speaker. We're hearing your conversation.
Okay. Yeah, thank you for letting me know that. Is that any better?
Can you try now? Let's see.
That's okay. There's no echo now.
Yeah. Anyone else who has their mic or a speaker, please.
Okay. Let me try again. So the central panel shows that these cardiosphere-derived cells that are called CDCs work not by stem cell mechanisms. They don't simply engraft in injured tissue, proliferate, differentiate into heart muscle. Instead, they lodge in the muscle briefly for maybe a week or two. And during that time, they secrete extracellular vesicles that themselves mimic the action of the cells. And both the cells and their secreted extracellular vesicles have a number of mechanisms of action that are quite intriguing. They're all tied together by the bottom one, immunomodulatory, but among the important mechanisms that made us think that they had a potentially broad therapeutic implications include the antifibrotic and the anti-inflammatory mechanisms. This discovery led to a number of clinical trials, all of which showed some indication of disease-modifying bioactivity. But as we learned more about the cells and we learned more about the potential indications, we focused on Duchenne muscular dystrophy. Duchenne muscular dystrophy has a cardiomyopathy that is actually the most frequently the fatal event in these boys and young men. It starts to appear typically after the skeletal myopathy is evident, usually within the second decade, and as I said, can progressively prove fatal. So this is now in front of the FDA for potential approval for Duchenne cardiomyopathy. The cells are given intravenously every 3 months, and the protocol that was described in the HOPE-2 paper, which was in the Lancet a couple of years ago, has been reproduced in the HOPE-3 trial, which has recruited 105 patients with advanced Duchenne muscular dystrophy. It's a randomized, double-blind, placebo-controlled registration trial. And stay tuned for the results, which should be available per the company that sponsored the work as early as later this week. Given that background, this talk is not about cell therapy, but rather about the once you recognize the actual mechanism of action, you can then focus in to try to define what it is that the extracellular vesicles are doing. And one place where we focus is on the RNA contents. The RNA contents seem to be important to us because the vesicles become inactive if you lightly solubilize them with some kind of a detergent that disrupts the contents and exposes them to ribonuclease, but doesn't degrade the proteins. The extracellular vesicles also don't contain any DNA, and they have very few mRNA transcripts. And that led us to focus specifically on non-coding RNAs. We first focused on microRNAs, which are the usual suspects. People like them because they know how they work. So we and others mined the contents of extracellular vesicles to see if some of the active principles might be microRNAs. And we got some hits, but really we were fascinated by the fact that there were a large number of totally obscure but very plentiful non-coding RNAs, and we wondered if any of those might actually be responsible for some of the bioactivity. So we synthesized, we sequenced the contents of the vesicles and synthesized the most prominent ones, and it turned out that so in that context, we found that several other things that we didn't suspect and we knew nothing about, including this pale green area here, which represents yRNAs, had members which stuck out like a sore thumb. And the single most prominent non-coding RNA within the inventory of RNAs in these vesicles was something we ended up calling extracellular vesicle fragment Y fragment 1, or EVYF1. And EVYF1 is a perfect homologue to the first 56 nucleotides of the human yRNA4 gene. And the human yRNA4 gene itself is 96 nucleotides long. EVYF1 was the perfect homologue to the first 56. The initial read had a sequencing error, which introduced an extra T, but upon resequencing, it was really a perfect reproduction of the first 56, 5' 56 nucleotides. So we wondered what this was. The whole yRNA itself is a bit mysterious. There's 4 yRNA genes in the human genome and about 30 or 35 pseudogenes. No one is quite sure of what they're doing. There's some presumption that they're involved in DNA replication, but the clinicians in the audience will recognize yRNAs as being the partners for Rho and La ribonucleoprotein autoantigens in lupus. In any case, EVYF1 lacks both the Rho and the La binding sites, so we had no idea how this thing might work or if it had any bioactivity at all. So we made it by solid phase synthesis. One advantage of RNA, small RNAs like this, is you can just use a machine to make a perfect copy. And when we did so and put this oligonucleotide into lipoprotein vesicles, actually just lipid vesicles, no lipoprotein, using just a simple transfection reagent mixed with the RNA, and we exposed those particles to bone marrow-derived macrophages, we found that they lit up the expression of IL-10, an anti-inflammatory cytokine, but a scrambled version of the same nucleotide content, but in randomized order, had no effect on IL-10 secretion. Likewise, in vivo, when we gave the RNA in the lipid particles 20 minutes after reperfusion, so this isn't pre-treatment, it's treatment after the ischemia and reperfusion, we were able to diminish the size of infarct size measured a week later by about 60%, but the scrambled version had no consistent effect. So we have discovered a novel natural cardioprotective non-coding RNA with an unclear mechanism of action. And even though it might be tempting to just develop the natural RNA as a therapeutic candidate, its structure was not compatible with either the FDA or the European Medicine Agency conventions. And what are those conventions? Well, first of all, it's startling how many RNA drugs there are, and with the exception of the mRNA vaccines shown here, all of these are non-coding RNA drugs. And the non-coding RNA drugs, by and large, are targeted to well-characterized disease pathways. They're small interfering RNAs or antisense oligonucleotides. There's one aptamer. And structurally, they're all 18 to 30 nucleotides long and chemically modified for stability and decreased immunogenicity. I should say it's a multi-billion-dollar global market with a cumulative annual growth rate of more than 14%. So reflecting on these structural kinds of conventions, we wondered if we could make a drug bio-inspired by EVYF1, something that looked to regulatory bodies very much like these 20-odd drugs that are already approved, and figure out the mechanism. So unlike the conventional pharmacopoeia in non-coding RNA drugs, where you go in and you target a specific disease pathway, we had no idea how the drug worked. We figured out a structure, and then we had to figure out the mechanism. So the first thing we did was to create a synthetic drug from EVYF1, and here's the EVYF1 sequence. We had an important clue where the action was, in the sense that another oligonucleotide that we called NT4 was very plentiful in the EVs, and it itself was also bioactive. So using NT4 as the 24 nucleotide template, we started making point mutations, and we started making chemical modifications. And you can see here some of the results of the point mutations and some of the results of the chemical modifications, and these are described in the legend over here. But let it suffice to say that after this structure-activity relationship optimization, what pharma would call SAR, we came up with a structure, TY1, that had a point mutation, so it was no longer a natural sequence. It was a G to C conversion in the first nucleotide of this oligonucleotide. And it had 6 backbone modifications known as locked nucleic acids, which stabilize the ribose bridges and make the structure relatively resistant to degradation by ribonuclease. We then took this sequence and this drug with its backbone modifications, TY1, put it in a transfection reagent and exposed those particles to bone marrow-derived macrophages and did transcriptomics. And you can see here in this display, which shows the changes in gene expression for any given gene and for one raster line, there's a lot of changes. When we look at gene ontology analysis to see which pathways are altered, you can see that the single greatest pathway with alteration is the DNA damage response, but there are many other things here involving macrophages and DNA. So we went and we went about trying to find out what was going on. How was TY1 inducing these changes? We worked on RNA protein interactions for about 18 months and found some hits, going along the presumption that this might be a ribonucleoprotein RNA interaction. And we got warm, but nothing held up to rigorous scrutiny. So we then resorted to RNA pull down with 2 methods, bait capture and probe-based capture, with the presumption that maybe this was an RNA interaction. And indeed, we got a hit with a molecule called TREX1 as a potential partner. And TREX1 was not something we were familiar with, but it turns out to be a super interesting exonuclease that's known as 3' DNA exonuclease. So TREX1 is an acronym for that. And what was interesting about TREX1 was both the fact that if you have loss of function mutations in TREX1, you get monogenic lupus, and loss of function of TREX1 has also been associated with several other autoimmune and inflammatory diseases. But also, when you look at TREX1, it's the gatekeeper for activation of innate immunity and the pathway involved in triggering innate immunity with CGAS, STING, which then turns on interferon signaling. What happens is that TREX1 normally degrades cytosolic DNA, which you'll appreciate is not something that the cell likes. The cell is uncomfortable with DNA in the cytosol. Primordially, this was probably attributed to a viral particle, and TREX1 indeed originated in unicellular organisms and has persisted since then. In mammalian cells, the presumption is that some kind of stress, possibly due to reactive oxygen species or other undefined disease processes, causes DNA damage, and that DNA damage leads to cytosolic DNA accumulation, which could parenthetically come from either the mitochondrial DNA or from the nucleus. TREX1 can normally degrade that DNA and prevent C-gas and sting, but when the accumulation of cytosolic DNA is so intense that it overwhelms the enzymatic capacity of TREX1, then you get activation of C-gas and sting. Gas and sting can be activated by other pathways, but this is one way of serving as a gatekeeper for activation of gas and sting. And basically, once interferon signaling starts and inflammatory cytokines are secreted, there's a danger signal to the entire body. And all of you who have suffered from, bacterial infections, severe bacterial infections, or severe viral infections can tell how it feels to have circulating interferons due to activation of innate immunity. So we conjectured that a compound that would increase TREX1 activity, we had no idea at the time whether it would increase or decrease or do nothing. We just knew that there was an RNA-RNA interaction, but we conjectured that a compound that increased TREX1 activity would have broad disease-modifying bioactivity. And indeed, when we put TREX1 onto bone marrow macrophages, you can see on this right-hand panel, the expression, the message levels, the mRNA levels of TREX1 increased about fourfold. A scrambled version of TRI1 with the same nucleotide content, but in randomized order, had no effect. And we narrowed down the effect to an interaction with the 5' untranslated region of the TREX1 gene. When we took the particles, and instead of putting them on bone marrow-derived macrophages in vitro, we put them into a rat myocardial infarction model 20 minutes after reperfusion, mimicking a typical MI in which the patient has an obstructed artery and comes in and has that artery opened iatrogenically. We measured infarct size histologically 48 hours later. We found that scrambled had no benefit, the gray stuff being tissue that's on its way to scarring. But TRI1 appeared to reduce the scar burden, and indeed, pooled data showed that scramble had no effect in the percentage of infarct mass in the ventricle, but TRI1 reduced that by about 50 or 60%. Likewise, circulating levels of the ischemic biomarker troponin I were decreased at 48 hours. When we looked at DNA damage in the cell, linking it back to the putative mechanism, and this is done by phospho-H2AX imaging, so it's a phosphorylated histone we're looking at. The vehicle and scramble were not that different, but TR1 appeared to be lower in terms of DNA damage, and the pooled data showed that that was true. And then if we looked at bulk left ventricular tissue, we found increases in the tissue of TREX1 levels after exposure to TY1, but not after exposure to scramble. This turns out to be somewhat surprising. It's in the right direction, of course, but we think the mechanism of action involves mostly macrophages. And so the fact that you can see it in bulk tissue was encouraging, but somewhat surprising as well, but certainly consistent with the proposed mechanism of action. So we dissected this. The paper will come out, I think, in a day or two in Science Translational Medicine. We presume then that TRI1 goes into macrophages, and the reason that we implicate macrophages is shown in the paper. But basically, if you deplete macrophages, there's no benefit. If you do adaptive transfer of conditioned macrophages that have seen TY1, you mimic the benefit, and so on. There's actually a whole list of controls that makes us pretty confident in this mechanism. Increasing TREX1 levels, and this increase in TREX1 levels blocks DNA damage, and in a way that is kind of self-perpetuating through a mechanism involving macrophage regenerative activities, in terms of basically the macrophages themselves are altered, but what they secrete is also altered. So it's not just a one-off in terms of a DNA molecule within the macrophage, but rather the secretome of the macrophages themselves altered. And altogether, there's a reduction of inflammation, fibrosis, and infarct size. So we wondered if this sterile inflammation model, which is what myocardial infarction is, would be predictive of broader bioactivity. We use it as a standard screen for bioactive molecules in the lab, but is it good just to tell us what's cardioprotective, or might it be a sentinel for other bioactive processes or targets for other indications? So we looked at another disease of fibrosis and inflammation, namely scleroderma and systemic sclerosis. It's an autoimmune disease affecting women in midlife. There's about 60,000 new cases a year in the United States, and we all know it from its presence in the skin, which is a very easy phenotype to detect. The deposits of scar tissue in the skin can be so severe as to cause contractions of the mouth and digits. But when it involves the internal organs, as it does in at least a third of the cases, and the heart and the lungs are particularly troublesome in terms of their involvement, that's known as systemic sclerosis, and that portends a 30% 10-year mortality. I became interested in scleroderma as a resident, having seen several of these patients die tragically and prematurely. And when I revisited the concept of, what other indications might we pursue, I thought of scleroderma, and I was somewhat disconnected with the general internal medicine literature at that point. And I said, well, this must have been cured by now. And instead, I found that there are no specific drugs or therapies that have been approved for scleroderma. One exception is a repurposed monoclonal that's been shown to be bioactive in idiopathic pulmonary fibrosis, which does slow the progression of the interstitial lung disease and systemic sclerosis, but doesn't seem to have any benefit on mortality. So we implemented 2 mouse models of scleroderma to see if 201 might help. This shows the results of one, and the idea here was to create a scleroderma phenotype by injecting subcutaneous bleomycin for 3 weeks. And this is a very standard model. And only after the induction of the disease did we randomize the animals to either receive vehicle, so that just shows you the natural history, scramble, the scrambled nucleotide version of TY1, or TY1 itself. And then we gave it twice a week, either intravenously or orally. Interestingly, we have an oral formulation. These data here are from the oral formulation, and what we find is that basically when we phenotype at week 7, so after 3 weeks of induction of disease, 4 weeks of treatment, and then phenotyping, we find that the skin thickness, control means normal animals that didn't receive bleomycin or drugs, the skin thickness is measured between the epidermis and the subcutaneous fat layer, is pretty thin. Vehicle shows a marked thickening of the dermis. Likewise, in scramble, no benefit whatsoever. But 201 appears to revert the dermal thickness back to normal levels, and in fact, this is evident in the pooled data. More exciting for us as cardiologists, when we look at a Mason's trichrome stain of the heart, which produces a brick red appearance for the normal heart and blue-gray deposits for fibrosis, we find that the fibrosis is not apparently altered by scramble, but is largely dissolved by TY1. And in fact, the pooled data confirm the lack of bioactivity of the scramble, but the bioactivity of the TY1 itself. And as I said, these data were confirmed in another model of scleroderma, the thin-skin TSK1 genetic mouse model, as well as in human macrophages and human fibroblasts from scleroderma patients. So let me just reflect for a minute on how this work that I've described differs from the conventional non-coding RNA drug development paradigm. In that paradigm, we start knowing a disease process. For example, here, how transthyretin accumulation can lead to amyloidosis. We design an antisense oligonucleotide or an siRNA to interfere with the synthesis of TTR, and we can then treat amyloidosis effectively that way. So the idea is, you know the mechanism, you define a target, and you make an RNA drug. We do it differently. We use a discovery-based paradigm in which we start with therapeutic cells that secrete extracellular vesicles. We mine the extracellular vesicle cargo to identify natural but obscure non-coding RNAs. We test those for bioactivity. And the ones that seem intriguing, we use to create bio-inspired chemical entities, which we call Xomers. That's a new drug class. And only then do the Xomers undergo mechanistic investigation. So an RNA candidate leads to a synthetic RNA. We test for bioactivity, and we explore the mechanism. And this whole pathway we call CRADO, cell-derived RNA from extracellular vesicles for bio-inspired drug development. I'll show you that it's generalizable. It's not just this one drug. TR1, though, is the Xomer that's most advanced in development. It's got an unprecedented mechanism of action. No one had ever thought of enhancing TREX1 activity as a disease-modifying principle. And as I told you, we have a formulation that makes it orally bioavailable, which was described in a paper published earlier this year in the Journal of Extracellular Biology. So what are the next steps to the clinic? Well, we've gotten pre-IND feedback from FDA last year. They fully accepted the efficacy data, but asked us to redo all the toxicology studies, which we had presented to them, homegrown toxicology studies under GLP standards with GMP grade drug, and those studies are underway with a view to us being able to file an IND by this time next year for sure, and with first in human studies to follow. I've hinted that this paradigm may be generalizable, and I've told you the story in which CDCs led to CDC extracellular vesicles. We mined those and identified EVYF1, which we now call WireX2, just for naming convention that I'll reveal in a second. But that created 2I1. But there's all these other things that have led to drug candidates, and in thinking about them, basically the nomenclature that we came up with was that there's this, after the extracellular vesicles is a row of natural non-coding RNAs. And we ended up calling those by the lowercase family letters, so M for message. Where did the annotation take us in terms of where they likely were? This is a fragment of a message. This is a fragment of a yRNA, a mirror. Trex is a tRNA fragment. The EX stands for exosome or extracellular vesicle. And BCYRN1 already is a long non-coding RNA that already had been named, so we're not going to rename it. And the sequential number, WireX1, WireX1 was EVYF1, WireX2 was NT4, so it's the second one that we characterized. And then the Xomers, the actual drugs, we call with a big letter T for therapeutic, the uppercase family letters of yRNA, and then number 1. So TY1 is this drug I've told you about. It was the first drug that we fully characterized. And now I'll tell you the story of TY2, which came from immortalized CDCs that made immortalized extracellular vesicles that we call IMEX. From that, we mined YRX3, which is a non-overlapping yRNA that was also very prominent in this population. And from that, we created TY2. And TY2 turns out to have an entirely different mechanism. But first of all, here's that yRNA4 in its 96 nucleotide length, along with a putative folding diagram. And you can see in the green the sequences that have been attributed to our previous inspiration for TY1, the NT4 sequence. But here in red is where this new thing is, the YRX3. And YRX3 we've characterized fully in this paper that appeared last year in the European Heart Journal. We find that it works in a very interesting way. Basically, it's totally different mechanism and a totally novel mechanism again. It inhibits the activity of protein interacting with C kinase 1 or PIC1, which then through a series of events eventually leads to enhanced macrophage efferocytosis as the central therapeutic principle. So it basically creates avid Pac-Man out of macrophages that we thought would clear dead cells in tissue and promote tissue repair. So we used YRX3 as bioinspiration. We went through a structure activity screening process to create the TY2 therapeutic candidate. This is what the SAR looked like. And what ended up being is, again, a point mutation of the natural sequence and also with six locked nucleic acid residues in the sequence to stabilize it and make it RNase resistant. So when reflecting on the mechanism of action of the compound, this increased macrophage efferocytosis, we wondered if it might be useful in arrhythmogenic cardiomyopathy. And arrhythmogenic cardiomyopathy, as many of you will recognize, is a monogenic disorder that involves one of several genes that are involved in cell-cell junctions. Desmosomes, these are genes like plakophilin and desmoglein. There's no disease-modifying treatment. You can put in an ICD and shock the patients out of the arrhythmias. You can prescribe less exercise, but there's nothing you can do that alters the natural course of the disease in terms of drug therapy. The problem is that they accumulate fibrofatty interstitial deposits, which leads to conduction block, arrhythmias, and in severe cases, dilated cardiomyopathy and heart failure. And basically, the mechanism summarized here appears to be that somehow, and I'm not sure how this happens, but the desmosomal defects lead to inflammation and fibrofatty deposits. Macrophages try to clear those, but are overwhelmed. So we reasoned that if TY2 upregulated macrophage efferocytosis, perhaps it would be more effective in tissue clearance. So as I said, that was the inspiring hypothesis. We then implemented a model of ACM it's a transgenic model in DSG2 mutant mice. This mimics a human disease mutation, which is quite aggressive in the desmoglein-2 gene. And basically, we started with animals at 4 weeks of age when the phenotype is already evident. They have ventricular dilation and inducible arrhythmias. Here is the echocardiogram, and here are the pooled data for left ventricular ejection fraction at the beginning of the study, in other words, at 4 weeks of age. And by then you can see that the ejection fraction is already in the 50s, and a normal mouse ejection fraction is about 70 or greater. So at that point, we randomized the animals to receive either vehicle, and this is given intravenously twice a week in this case, vehicle, in which case the natural history revealed a progressive decline of ejection fraction over the following 6 weeks, or TY2, which led to a restoration of ejection fraction, not all the way back to normal, but trending very close to normal. When we looked at the tricuspid annular excursion as an indicator of diastolic function, we found that it was improved by TY2. Electrocardiographically, in the basic EKG, there was an abbreviation of the QT duration by TY2, and the corrected QT was even more evidently affected. But when we induced ventricular arrhythmias in these mice, we found them to be almost uniformly inducible in the absence of TY2. But after TY2, there was a marked decrease in inducibility. The significance level in these data was not yet significant, but we've subsequently done more experiments and found it to be a consistent and significant reduction. This is as yet unpublished data. So TY2 is an Xmer. It's bioinspired by another non-overlapping yRNA4 fragment. It's also cardioprotective, but has a totally different mechanism of action, which is again novel. The mechanism of action suggests there might be utility in diseases marked by interstitial junk, for lack of a better word. It's also orally bioavailable. I haven't shown you those data, but we have demonstrated that. And we're entering the regulatory pathway with TY2 with a view to pre-IND and IND, and we're developing it in this case for the ACM indication. I'm not going to tell you about the other branches of the tree that have already evolved, but we, just to mention the generalizability, extracellular vesicles made from induced pluripotent stem cells have led to the recognition of an endogenous interesting sequence, MREX1, which is cardioprotective, and we're using it as bioinspiration for a new class of drugs. Told you about TY2. T-Rex, which is a fragment of a tRNA, has led to the creation of a synthetic drug TT1 that we're developing for a variety of skeletal muscle diseases. And then BCYRN1, which is a long non-coding RNA previously described in the brain. We find the long non-coding RNA itself, when overexpressed systemically, leads to upregulation of regulatory T cells, with an increase in their proliferation and in their activation. We figured out the mechanism and made 3 small oligos that mimic the benefit of BCYRN1, calling these therapeutic long non-coding RNAs 1 to 3 or TL1 to 3. So we're using this paradigm to create new drugs. How it really happened, I've shown you, but let me reflect finally in concluding the steps in the discovery arc, what the conceptual innovations might be for those of us who think about extracellular vesicle signaling in particular, and some of the practical consequences. So we began more than 20 years ago with an attempt at cell therapy targeted at the heart. We then identified extracellular vesicles as the key mediators, and we began to mine the extracellular vesicle contents to identify defined factors, and that's how we found EUYF1. We then created bio-inspired new chemical entities, TY1 and TY2 being examples. We're now exploring new extracellular vesicle cell sources, not limited just to CDC EVs. And mechanistic analysis of the drug candidates has led not only to unexpected biology and some new disease-causing pathways or disease-modifying pathways, but also indications that we might never have foreseen when we started this journey a couple of decades ago. And this credo paradigm has yielded new leads and new drugs. That's ongoing. Conceptually, the fact that extracellular vesicles contain all these non-coding RNAs with apparently complementary mechanisms teaches us that the biology of extracellular vesicles is very rich and complex, and potentially all these fragments are working synergistically, and that might help explain why these extracellular vesicles themselves have strong disease-modifying bioactivity. Many of these EB-derived non-coding RNAs appear to act on macrophages. I haven't shown you that data, but we feel that that's sort of a grand central station of the mechanism of action of these drugs. And drugs that act that way on macrophages can be delivered orally in this formulation that we've described, involving casein and chitosan micelles. The sterile inflammation model, myocardial infarction, turns out to be highly predictive of broad therapeutic efficacy, not just ischemia reperfusion. We find that the disease-modifying bioactivity in vivo, not only in MI, but in a bunch of other diseases, is notable because it works after injury. It's a reversal of established disease, not prevention. And so it's obviously of greater therapeutic utility than in something where you have to identify patients at risk and treat them beforehand. Practically, we've filed 41 patents on this RNA estate. They're all relatively recent, but one's already been granted. And we have 7 proprietary therapeutic candidates in the pipeline. 3 are at the proof of concept optimization level. 3 are already in development for regulatory filings. 2I2 for ACM, TT1 for Duchenne muscular dystrophy, and we're also developing 2I1 for HFpEF. We have a publication showing that it's bioactive in a HFpEF model. And 2I1 is in the most advanced in IND enabling studies for scleroderma, as I've shown you before. So if I want to conclude by acknowledging the folks who did the work, Ahmed Ibrahim has been a partner from the beginning. He was my PhD student. First identified the role of extracellular vesicles as mediators, and has stayed on after a false start in the biotechnology industry. He came back to academia and has collaborated with me in developing the drugs that I've discussed. Alessandro Chulo was primarily responsible for the YREX3 and TY2 work. Special thanks to everybody here and for funding from these sources. CIRM, I should mention, is the California Institute for Regenerative Medicine, the peer-reviewed medical research program of the Department of Defense, has been helpful in providing the kinds of money. Both these agencies have been helpful in providing the kind of drug development money that NIH is loath to provide. Mechanistic studies, NIH.
Thank you, Eduardo. I think you may have cut out, your audio may have cut out. Can you hear me?
I can hear you now, yeah.
Okay, now you're back. Yeah. So I'm gonna funnel questions to you from the chat. You can also open the chat, but you may be busy right now. There's still a lot of people on the Zoom right now. 40 people or so are on the Zoom. Let me start by asking you, how does the first long non-coding RNA that you talked about work in the macrophages? How do they signal to reduce infarct size? Do they signal to the cardiomyocytes, or is it that they're eating debris that's in the extracellular matrix? How does it work?
So I think when you say the first one that I was talking about, you mean the 2I1 and its natural precursors. So our understanding right now is that when we deliver these drugs exogenously, they're taken up by macrophages, and that seems to be an absolutely necessary event, because if you deplete macrophages either with clutterated liposomes or with mafia mice genetically, you undo the benefit of TY1. So macrophages are required. They also seem to be sufficient in the sense that you can take bone marrow-derived macrophages, condition them ex vivo with TY1, wash off the TY1, put in those conditioned macrophages by adoptive transfer, and they mimic the benefit of the drug. So we believe that the initial event is uptake of the drugs by macrophages, kind of a phenotypic conversion or a reprogramming of the macrophages to be, very anti-inflammatory, not typically, can't pigeonhole them into a classic M1, M2 phenotype, but they have kind of a distinctive anti-inflammatory phenotype. And then that benefit is basically amplified by the fact that macrophages that have received TY1 themselves alter their secretome so that their own extracellular vesicles are themselves salutary. So macrophages are themselves modified to become more disease modifying, and then the signal is amplified by altered macrophage signaling.
So can you take the conditioned media from those macrophages and administer that and get the same reduction in infarct size?
Yes, we can. Well, more specifically, if we take the extracellular vesicles from macrophage conditioned media, they mimic the benefit. So the signal event appears to be drug uptake by the macrophages, then that signal is amplified, which may explain why the drug effects sometimes are rather long lasting, because they're not limited to the initial modification of the macrophages, but rather also dependent on amplification of the signal by the macrophages.
Thank you. I think Dr. Garcia wanted to make a comment. Mario, your audio may not be on right now. So I love this concept of finding the microRNA first and then working out the mechanism last, because the mechanism takes all the time.
Yeah, we now have gotten pretty good at picking out non-coding RNAs we like and doing the SAR and the finding a candidate. But that may take a year or 18 months, but what takes 3 or 4 years is then figuring out the mechanism. And of course, we're totally shooting in the dark. So we not only, in some cases, we have to go the ribonucleoprotein route, in others the RNA interaction route. Sometimes transcriptomics as a first step gives us some hints as to where to look.
Okay, I think we have one comment here. This is a great demonstration of the importance of RNAs and exosomes. If you do the opposite experiment and destroy the RNAs, what is the relative effect of proteins from exosomes?
Yeah, good question. So we've done that by lightly solubilizing the population of EVs just enough to expose the contents to RNase, but not to degrade the proteins. And that preparation loses 85% of its bioactivity. So the proteins may be doing something, but they're not able to explain the vast majority of at least the bioactivity in cardioprotection.
And we have a question from Leandro. Outstanding lecture and congrats again for an amazing and very inspiring career. My question is, one thing it always called my attention to is how quickly CDCs and EVs disappear from the heart, yet the benefits last for months. Do you think there is a future where we could use something like CRISPR to get the myocardium itself to make those helpful signals for longer instead of relying on transient cell or exosome delivery?
Yeah, that's a great point, Leandro. I mean, in general, if we were to, in a very general sense, if we were to understand mechanisms of drug action and be able genetically to edit the the responsible pathway, we could in principle make a permanent modification that's disease altering. And of course, this is the pathway that's being used with PCSK9 and other elements in the cholesterol and triglyceride biosynthesis pathways. So the natural progression there has been interesting, right? Small molecules, monoclonals, and then siRNAs or ASOs, and then finally gene editing. The problem with gene editing, of course, is in a disease like myocardial infarction, you might want to mitigate a pathway for a while, but you don't want to, necessarily block that pathway forever. But there are certain diseases where, particularly genetic diseases, where that would be a definitive correction.
Are there other questions for Dr. Marbán? Well, I'd like to thank you again, Eduardo. Very interesting and, as usual, incredibly well presented talk. Thank you for your time.
Thank you all for your